Hey guys, this will be our second A-level biology P5 video, and this time again I will be focusing on the first question. Perhaps we will do the second one as well if time allows it. And this paper is basically paper 5, planning, analysis and evaluation, and this really beautifully sums up all the skills you need to do this paper effectively. And this paper has a total of 30 marks, and you have to do this in 1 hour 15 minutes, so careful organization and distribution of time is a necessity. So let's start with the question and you really need to read out the question carefully and make notes along the margins. They're really helpful in you getting a visual of the whole scenario you need to do all those questions on. So a student investigated the effect of different growth media on the germination and the growth of pollen. So what? Number 1, different media. Number 2, germination and growth. And immediately you have an idea what the question will be asking. And the growth media tested are shown in table 1.1, and this is table 1.1, and we have 1, 2, 3, 4, 5, a total of 5 different media that we will be investigating. We have a total of 1, 2, 3 different media we will be investigating, and how these media are different are also expressed in the table. There are 5 essential components and their concentration within the media is written. So you can tell that media A is just sucrose solution of basically a solution of 10% sucrose, so 10% of the and then there's medium B and 10% sucrose solution, and it is also contains 100 milligram per decimeter cube boric acid and 300 milligrams per decimeter cube calcium nitrate. And for medium C, again it's 10% sucrose, and this is something that is the same, again 100 and 300 the same. And the difference is that when we compare B and C, we see we also have magnesium sulfate and potassium nitrate and the concentrations given which are absent in B. So again, keep the differences in mind. And figure 1.1 shows the apparatus used by the student in this investigation. Again, what is the apparatus? We have this petri dish, and I guess it's a petri dish, and we have a glass slide and we have supports for it, and underneath the slide we have water, and on top of it we have pollen mixed with the growth medium, either A, B or C, and we have basically covered the petri dish, so something that does not belong, that is in the air, does not get into the pollen and growth media mixture. And the student carried out the following procedure when investigating each growth medium A, B and C. If we have 3 different media, obviously we will be investigating all 3 of them. And what was the procedure followed? So pollen from a mature anther of a flower was removed with a paintbrush. It's simple, you just simply touch the anther with a paintbrush, some of the pollen will become dislodged from the anther and adhere to the paintbrush. And the pollen was mixed with 2 drops of the growth medium, so 2 drops of either A, B or C depending which one we're investigating at the moment. And the slide was placed in the petri dish on top of slide supports with water underneath, and this apparatus was kept at room temperature for some time so that it becomes equilibrated, and every 10 minutes the slide was removed and the pollen was observed using a light microscope. So at intervals of 10 minutes the slide was observed using a light microscope, and the percentage of the pollen germinated was investigated. And the length of the 10 longest pollen tubes in a field view were measured, and the mean pollen tube length was calculated. So number 1, the first step has to do with the acquirement of the pollen. The second step was basically the key of the whole investigation, the mixing of the pollen with the whatever media we were using. The third has to do with the apparatus arrangement, and the fourth step is basically the whole concept of keeping the temperature constant for the investigation. And the fourth is basically when we mixed it, and we would expect the pollen would have started germinating because it has some of the conditions needed. And so you will be investigating how much germination has occurred, how much growth of the seeds, whatever. So what would you observe? A pollen is basically a bit of a round structure, and you would observe a lot of them. There would be a hell of a lot of them on the slide, and they would be spread out. Alright, and a simple ungerminated pollen would likely be circular. However, for germinated portion there would be a tube-like structure that would be emerging from it, and you would basically be looking for that every 10 minutes. And the percentage of the pollen germinated was estimated. How do you do that? Well, you really can't count each and every one of the pollens on the slide, because hell, there would be a lot of them even on one slide. So estimation would be that you would take 10 different parts of the slide at at a say 400 magnification, and all of the pollen that are like visible, you just count all of them up, and then you count the ones that have germinated or shown visible signs of germination. Say the pollen tube is visible, and you basically divide those from the one, and you do that a couple of times, and you choose the part of the slide that you're investigating randomly, and then just you get a mean. And then length of the 10 longest pollen tubes in a field were measured. So in a particular randomly chosen field, you observe a lot of tubes, and the one which you think are the longest, you basically measure them up, and then you basically take the mean. And for the measurement you have all those graticules and micrometer slide and stuff. Okay, this is basics. And the results of the 3 investigations. And 2 different parts of the investigation, and the mean length of the 10 longest pollen tubes after 10 minutes, after 20, 30. So we have intervals of 10 minutes, and we have 1, 2, 3, 4, 5, 6, 7 different events at which we took basically the slide. And you can see, you can observe at the same time interval the growth in A, B, and C, and with time, same for here. So what is the independent variable? That would be the medium used, or the identity of the medium, whether it was A, B, or C, or perhaps it could be the concentrations of the 5 components, and the dependent one would be the one that is affected, and we could say that was the mean length of 10 longest tubes after a time interval, and the percentage germination after a given time interval. Describe how the student could have made the length of the pollen tube using a microscope. From AS Biology, you would be aware of 2 things. Something called an eyepiece graticule. And that is something that is like in part of the whole microscope. And another thing is the stage micrometer. And the stage micrometer is actually on the the stage. The eyepiece graticule, on the other hand, is basically on the eyepiece, and you basically use these for calibration of the eyepiece graticule, and then you use them to measure the length. Oh, over here, right? They would have made use of an eyepiece graticule, and which would be calibrated using a stage micrometer, and they could have basically used them to measure the length, while the percentage germination was calculated, because you'd be taking different samples. For example, a particular region of the slide under the view of the microscope, and the number of pollens per view would not be the same, and that is why we take percentage germination to take into account the fact the number of pollens per view is not the same. And student carried out t-tests on the results to compare the length of the pollen tube when grown in different media. And to carry out a t-test, the number of the sample must be counted. It is also necessary to carry out 2 calculations that must be carried out. Number 1, the mean of each set, and the standard deviation of the mean of each set. And since the student decided to use 18 degrees of freedom in each of the t-test calculations. Well, how do you calculate the degree of freedom? It's basically, the formula is n1 minus 1 plus n2 minus 1, and n1 is the number of samples for the first set, and n2 is the number of samples for the second. So how many samples are there? 1, 2, 3, 4, 5, 6, and 7. Right? I'm sorry. 10. Basically 10 would be for the first one and the second. So 10 minus 1 is 9, plus 10 minus 1 is 9, so 9 plus 9 is 18. And the student carried out a number of t-tests to find out if after 70 minutes the difference in the mean pollen tube length is significant. And the value t is equal to 2.5, t is equal to 3.56, t is equal to 1.94, and the critical values are shown. And the degree of freedom which we should be using ought to be 18. And 18. And for the DP, you should remember that the probability increases when the value of t decreases. So for 18, it has to be 2.1 or any value less than 2.1. So A and B definitely significant. A and C also significant. B and C, however, is insignificant, and in the last third scenario, the null hypothesis is valid. and said what conclusions can be drawn about the significance of the differences in the mean length from the three values of t given above. Simple interpretation, you just need to have a grasp of the statistical tests and how to interpret them, which is actually the most important part. Students somehow often miss out on. And from the results in table 1.2 in the t-test, the student concluded that calcium has no effect on the germination of pollen grains, but increases the growth rate. And sucrose is needed for pollen germination but not the growth rate. And we have to give references to table 1.2, anything that suggests these, that supports them and does not support them. Let's look at these as well as T1 and T2. And for that, look over here. A has no calcium nitrate. B has 300 and C has 300. And when you look over here in the case of germination, here first, if you compare A with B and C, you get that there's a difference, but it's a bit confusing. And then when you look over here, you see that there is a difference there as well. The mean length 23.9, 27.6 and 38.2. And you get the idea that perhaps the fact that calcium is present is in fact having something of an impact. And you're like, yay, great. But that is not the whole of it. You need to also take a look at the t-tests. The difference between B and C isn't insignificant. It's insignificant, so you may assume that they have the same conditions and any difference is merely due to chance. And then you can say that indeed calcium nitrate is having an effect on the growth rate, because the growth rate of both B and C is greater than that of A, and any difference in B and C, and there is a difference, is merely insignificant. And this means that they have literally the same conditions, and that the differences in the components, the two things that are different for C than B, they don't really have any part to play. And therefore, because both of them have a greater growth rate than A, then yep, significant. However, when we talk about the A part of it, for the sake, we have to see that there isn't any effect on germination, you'd be right. Because we're saying the difference between B and C, because we're only, this T is about the mean tube length, right? And so when we talk about the germination part of it, there isn't any pattern, is there? There isn't any pattern there, because B is greater than A but C is less than A, so we may assume that there is no correlation between them, or at least that there are some other factors that are also influencing it, because there are differences between B and C as well, and that we cannot say if any difference is due to calcium alone. Again, one would be validity and C would be the second part with the reference to the germination. We cannot be sure because we haven't performed any t-test, and we cannot say that any difference between B and C, and there is a difference, is in fact influencing the result, and it isn't due to calcium. So there may have actually been some correlation between germination and Ca2+, but we cannot observe it because there isn't conditions for fair comparison. And then there is sucrose. Sucrose is literally the same for all of them, 10, 10, 10. And let's check out the fact that for A, B, AB, it is significant. AC, it is also significant. For BC, it is not significant. So we have to compare those. Where are they? Yep, over here. So B and C. And we're saying that the difference in the mean length of the 10 longest pollen tubes, there isn't any difference, and we're saying that the conditions are the same. Any difference is merely due to there being chance. And if you talk about A and B, we are saying that there is any difference in the mean tube length is significant, meaning that the conditions are different. So you really can't say whether sucrose has any effect on the percentage germination or the mean length, because the sucrose is the same. And if we're saying, especially in the case of the mean length of the test 10 longest pollen tubes, in case of A and B, we shouldn't be able to make comparisons. And we have to outline a further investigation that the student might carry out to provide more support for the conclusion about the role of sucrose in the germination of pollen and in the growth of pollen tubes. We could basically standardize the conditions that sucrose is the only thing that is being different, and then we could compare it and predict the results of this investigation if the student's conclusion about the effects of sucrose is correct. The conclusion was that it is needed for pollen germination, but has no effect on the growth rate. So what we would get as a result would be that germination would be affected. Initial portion of it, we would observe that the time for it would be less, but not so for the case of the lengthening of the shoot that results. Now take 5 minutes and go over this question and see what your thoughts are. I'll be pausing. So hopefully you've gone over this. Let's now take a look at the marking scheme and the notes I've made about this and discuss this in detail. Yep, here it is. Number 1, independent is the composition of the growth medium. Again, which of the growth media it is. This is better way to write it though. And the dependent variable is the length of the pollen tube or the percentage germination. So mean length of the pollen tube and the percentage germination. 3, reference to suitable magnification from 40 to 400 vex. Yep, use of eyepiece graticule 1 mark. Calibration of eyepiece graticule with micrometer 2 marks. And to find the value in millimeters of an eyepiece unit 2 marks. And conversion 3 marks guaranteed. This is basically theoretical. Just take a look at the topic about it in AS. Idea of allows comparison to be made between samples with different starting number. Yep, 1 mark guaranteed. Mean of each sample and standard deviation of each sample. Another 1 mark. Reference to 10 in each sample and subtracting 1 from each. Another 1 mark. Reference to critical value. Probability is greater than 0.05 is 2.1. And A plus B and A plus C both have values greater than the critical value, and B plus C on the other hand has a value less than it, and therefore A plus B and A plus C are significant and not due to chance, and they're therefore caused by an environmental factor. And reference to A plus C being also significant at, yep, so 3 marks guaranteed. 4 off. The pollen tubes grow longer better with calcium in the media in the case of B and C, 1 mark. And reference to pollen germination is almost the same with and without calcium in all media, or reference to pollen germination occurs with sucrose alone. Reference to boric acid causing an effect on pollen tube, yup, 2 marks. Reference to medium C has magnesium, potassium sulfate that may be increasing the pollen tube growth. Yep. Reference to all media have sucrose, so no control without sucrose. Yep. So if you had written what we were discussing, you'd likely have scored all 4 marks. 1 for this. Sucrose is present, not absent in any. Boric acid could be causing an effect. 2 marks. Medium C also has magnesium, potassium sulfate that may be increasing the pollen tube growth. 3 marks. Reference, yep, skip this, is almost the same with and without, yep, 4 marks. We are looking for an idea that presence of calcium has little to no effect on the pollen tube germination. Because you can observe here 18, and there's an decrease over here, and a further decrease again. Here there was an increase, again decrease. So there are other factors, but you don't really know which one we should be considering. And reference to making another growth medium without sucrose, yes, and making sure that all of the other conditions are the same. Idea of repeating the rest of the procedure for both germination and idea of pollen. I guess 1 mark for this. For pollen germination, there should be little to no germination, and there should be that the pollen tube growth ought to be similar. Let's take a look at the question. I actually forgot the question for the last part. Conclusion about the effect of sucrose is correct. Needed for pollen tube germination but no effect. If you don't have any sucrose, there would be little to no germination. And because we're saying it doesn't have any effect, so I guess from 19, if you've written this, you'd have gotten at least 16 marks. That's not bad, honestly. Let's take a look at the examiner report now. So candidates ought to have experience of practical work, familiar with the use of a range of equipment, recognize the key stage of an investigation, application of knowledge to interpret complex data, experience processing data, making decisions about conclusions. Most candidates identified the independent variable as a composition of the growth medium, though few thought it was time or temperature. Likewise, the two dependent variables of percentage germination and growth of pollen tubes were usually given 2 marks. Few mentioned that the microscope would be used at a magnification suitable for observing pollen, obviously, where practical experience at calibrating an eyepiece reticule should have allowed the candidates to approach this question with confidence. Eyepiece reticule was used to measure the pollen tubes and was calibrated using stage micrometer and not the reverse. You'd get a value in millimeters and then you'd convert it. 3 marks. The key idea was expected here was that percentage germination allowed a comparison to be made between samples, and the better responses made this point clearly. 1 mark. And the majority of answers correctly identified the mean and the standard deviation where the calculations needed. And the commonest incorrect response was to cite standard error. Some had chi-squared test in mind. What the hell? Expected and observed. Variance is basically the standard deviation, except you haven't taken the other road. Good answers were able to explain the idea of one less degree of freedom than the number of categories, and thus came up correctly with 18. Lack of understanding as to how this was calculated. 3 categories should be compared at the same time in the t-test. Obviously, you can only compare 2 sets. And it wasn't just enough that you had to give the formula, you'd have to explain what n was. Some centers are now clearly teaching the statistical technique successfully, and there were some well set out answers. Work at the 0.05 level, and the first thing to mention here is at this level and with 18 degrees of freedom, critical value is 2.1, and the first 2 values of t are higher than this, and thus these differences are significant. Third is lower than this and therefore not significant. And some had the idea wrong way around in thinking that 2.5 and 3.56 were not significant. So values are either significant or not significant, and there isn't any degree of significance or insignificance. And there were some very long answers to this, often spreading beyond the allocated. Overall pattern as shown at the end of the investigation. Not only data codes, you have to describe them. 3 pieces of evidence grow longer in media with calcium. Germination is similar with or without calcium, and that sucrose alone allows germination. Not the same as saying it occurs in all 3 media. Media have no sucrose, so there is no way of knowing its effect. How boric acid may be involved, and in medium C, so might magnesium, potassium, or sulfate. And the majority of candidates suggested doing a range of sucrose concentrations with no sucrose, but this was credited as part of a range if that was the way. Would we repeated this before? Yep. So 2, 5, 6, 7, 8, 9, 10, 11, 12, 13 and 14, 15. So out of 19 you'd get 15. Seems good enough. Hopefully this was helpful.